All right, so now on to our last major chromosomal rearrangement, which is translocation. And just as it says, translocation is really the movement of a segment of a chromosome, but to a non-homologous chromosome or another region of the same chromosome. So this is not to be confused with crossing over. Okay, so translation or translocations is where we see genetic material actually breaking off and joining with a completely different non-homologous chromosome. And that action this example is a Robertsonian translocation. We'll look more in depth at it in just a minute. But what it ends up resulting in is one large kind of normal metacentric chromosome and then a fragment of another chromosome that's usually lost. All right, so translocation, please don't get confused with it and crossing over. So crossing over is really referring to the exchange of genetic material between homologous chromosomes. So again, that's going to happen in prophase 1. That's like chromosome 1 crossing over and exchanging genetic information with the other copy of chromosome 1. Okay, so translocation is where we have genetic material leaving chromosome 1 and going to chromosome 5 potentially. So, two major types of translocations, reciprocal and non-reciprocal. Non-reciprocal translocation is where the genetic information or material is going to move from one chromosome to another, and there's not going to be any reciprocal exchange. Okay, so that can be visualized in this example here in the middle of the slide. So again, we've got our our two genes on this specific chromosome A and B are centromere, and then genes C, D, E, F, and G. And then if we were to see a non-reciprocal translocation take place on a completely different chromosome, represented here by MN, centromere, OP, R, and S, what it would look like is a portion of the first chromosome, and more specifically the genes E and F, would leave that chromosome and travel to the second chromosome and insert itself after the P gene. So reciprocal translocations are the opposite of that. That's where genetic material is going to be exchanged between non-homologous chromosomes. Okay, so not the same chromosome, but there's going to be an exchange taking place. It's not just genetic material moving from one chromosome to another and then nothing else taking place. We actually see a reciprocative event where genetic material from that second chromosome goes back to the first chromosome. And you can see that in the example here in the center of your slide as well. So we actually have genes or genetic material actually switching places between two different non-homologous chromosomes. So translocations can affect phenotype a couple of different ways. First of all, it can create new linkage relations that could affect gene expression. So this goes back to that concept of position effect. So if a gene is required to be in a certain position in order to be turned on or turned off, then we could be compromising that with some sort of translocation. If genes are moving to a new location and so they may have the ability to come under new control of different regulatory sequences or other genes. So if we do see that exchange of genetic material take place, realize that it could still work and be upregulated and turned on. It's just going to be regulated by different sequences or genes. And then another effect of translocations on phenotype is that we may actually see a disruption in a gene sequence. So if the genetic material that is broken off in a non-reciprocal translocation actually occurs in the middle of a gene and does not encompass the whole gene, then that gene most likely will not be functional because all of the genetic information is not there. So let's look more in depth at a Robertsonian translocation. And this is where two long arms of an acrocentric chromosome are going to be joined together, and they're going to be joined through a common centromere. Okay, so we've got two long chromosomes that have a very long arm and a short arm around the chromosome or around the centrosome. They're going to be joined together so that the result is going to be one metacentric chromosome and then a fragment chromosome. And usually that fragment is going to fail to separate or segregate, completely decreasing the total chromosomal number. So that has a lot of implications. So if we look at an individual that's represented by this figure on this slide, and this is a reciprocal translocation that's taking place. We've got a normal chromosome number 1 in red, a normal chromosome number 2 in blue, and then we've got a another set of chromosomes that have a translocation taking place. So we've got translocation chromosome number 1, translocation chromosome number 2. So what ends up happening is during metaphase, these chromosomes are going to try to pair up with one another, and they're not going to be able to do it in a normal fashion. So they form this cross structure because the chromosomes are actually having to bend so that they can pair up with the other set of chromosomes. So these are weird kind of synaptic structures. And as we move out of prophase and metaphase during the pairing and lining up on the metaphyseal plate into anaphase 1, we can see that we have a couple of different scenarios that could take place as those chromosomes separate and move towards the poles of the cell. So we could go through or could have this situation take place where N1 and N2 are going to segregate together and T1 and T2 are going to segregate together. Or we could have N1 and T2 segregate, followed by N1 and N2. And then the third combination or third chance is going to be where we see N1 and T1 segregate off to one side together, and then T2 and N2 segregate together. So as we progress through each one of these three possibilities into anaphase 2, we get very, very different results as far as gametes. So in the first example, if we have N1, N2 segregating, and then T1, T2 segregating, this is as normal as we can get, and it's going to actually give us viable gametes. Okay, so we've got N1 and N2 are going to be normal chromosomes, nothing weird about them. N1 and N2, the second copy, is going to be normal again. And then T1 and T2 are going to have exchanged genetic information, but it doesn't involve the centromere at this point. Okay, so they are able to go through, be copied, and segregate normally. So they result in viable gametes. In the second scenario where we see N1 and T2 segregating and then T1 and N2 segregating respectively, we get a whole lot of junk. Okay, so these gametes are not going to be viable at all because they paired up the wrong way. So we see that we've got a normal N1 and then we've got a T2 that is not normal at all, and they are trying to pair up with one another. And then the third possibility was where N1 and T1 segregated together and T2 and N2 segregated together. And again, once we go through anaphase 2, this really results in non-viable gametes as well because that genetic information is exchanging, but it's not pairing up correctly. All right, so what does all this mean? Well, this really has a lot of implications when it comes to some of our organisms that are very closely related to us as humans, specifically orangutans, gorillas, and chimpanzees. They all have 24 pairs of chromosomes, or a total of 48 chromosomes. However, in order for them to have evolved into humans, what we know through G banding pairing in evolutionary biology is that two chromosomes actually underwent fusion, end-to-end fusion, and that reduced the total number of pairs of chromosomes down from 24 to 23. So that fusion that's taking place here is illustrated in this figure. So we've got two acrocentric chromosomes that are actually going to be fused together into a dicentric chromosome. And then at the centromere, the second centromere eventually is going to become inactive, and that is going to result in our chromosome number 2. So that's how geneticists believe that we evolved from orangutans and our primate friends to humans. It's kind of cool. And we know that because we have a lot of genetic material that is similar to chimpanzees, gorillas, and chromosomes. So those chromosomes are shown here. And the banding that we see, the gray banding, even though it's pretty light, that is common genetic material that we see. So we can see we have a lot of relationship to chimpanzees, gorillas, and orangutans in this first set of genetic information. And then we see two other, or 1, 2, 3, 4, 5 different other commonalities between the primates as well as us. And this is just for chromosome 2. So just think about how many more pieces of chromosomal information on the other chromosomes relate back to these primates as well. All right, so rearrangements really become important in trying to diagnose and understand cancer. So if we see deletions that occur in regulatory genes, so genes that are like housekeeping genes, their job is to turn on and turn off specific cellular pathways, and we see deletion of that gene, they can actually run rampant and out of control. And that's where we start to see a lot of tumors being cancerous, is because those pathways have been shut off or not regulated. So translocations and inversions can bring two different genes together and can actually really stimulate cancer proliferation. And we see this with myeloid leukemia. And unfortunately, 90% of patients with this type of cancer have a reciprocal translocation between chromosome 22 and chromosome 9. So there very much is a genetic basis to myeloid leukemia. It's hard to treat because it is genetic. All right, so one more comment about chromosome structure before we jump into the rest of the chapter in the next video, but is fragile sites. So fragile sites on chromosomes are constrictions or gaps at a very specific location. And these gaps enable chromosomes to actually break easily. So one example that we see in humans is actually on the X chromosome. So the chromosome that's specific to females. And so one of the conditions that we have is called Fragile X syndrome. And if that fragile site is actually expressed, then we can see severity related to intelligence, and it results in individuals that are mentally retarded. So this can also play into a lot of behavioral issues as well.